mouthful.
Please welcome Eyal.
Thank you very much. Hi. So yeah, Marco, I do agree that my title, Protein Design by Multi-Objective Optimization, is quite a mouthful. And I set to myself a goal that by the end of these 30 minutes, you'll have an understanding of what I mean. In order to do that, I set up like 3 takeaway messages for you to take home. The first is I'll describe what multi-objective optimization is in general terms and why it yields better results than when optimizing for what I'll define as one-dimension optimization. The second takeaway is I'll give a list of characteristics that, I'll give you an intuition for when you can apply it, meaning that you think of your own projects that you're working on. Is this relevant for you? And the third, I'll show how I'm a data scientist in a biotech company called LabGenius, and I'll show how we apply machine learning and multi-objective optimization in order to improve the efficiency of how therapeutics are being discovered. A few more words about that. Today, we live healthier lives than in the past due to advancements in therapeutics, but the way in which new drugs are being discovered is it's much more risky than it used to be. It's much more expensive and takes much more time for the reason that many candidate molecules, they fail in various stages in the R&D and testing pipelines. The reason is that there are many characteristics that proteins have to pass successfully, and it takes only one parameter for it to be rejected. And R&D and testing cost a lot of money, millions and billions of dollars, and sometimes it takes pharmaceutical companies years until they find the pain points where it might fail. This is an example of big pharma company from 2016, in which they thought that they had an antibody that could reduce cholesterol levels, and only after a year-long third stage test did they realize that it lost efficacy on patients, and then they had to stop testing the drug, and eventually they had to retract claims in which they thought that they had forecasts in which they thought that they would have $500 million in revenue. And for this reason, in LabGenius, we are working with partners in order to make the process of figuring out which proteins are most likely to succeed more efficient. And we're doing so by means of machine learning and multiple objective optimization, the topic of this talk, as applied on empirical data of proteins that we're synthesizing in our laboratories. So the first part of this talk will not be about biology at all. It will be generic about multi-objective optimization, in which I'll describe what it is and how we can apply it with, in particular, with genetic algorithms. And then I'll talk about how at LabGenius we apply this for protein design for the sake of therapeutic discovery. Hi, my name is Eyal. I've been working in LabGenius for the past year and a half. I am a physicist by training. I transitioned to data science 5 years ago. And this is the point to thank the organizers of PyData London throughout all the years for creating this environment of meeting people and sharing knowledge in Python and in data. And I'm glad that I can contribute something that I learned in this past year. One last introduction slide. I work with an interdisciplinary team, and I just want to highlight 2 true unicorns that I'm very fortunate to work with. Mina just finished a half-year internship with us in which she worked both in data science and software engineering. And Katya, she is an expert in machine learning in biological experimental data. Okay, so with that intro, I'm ready to start. Optimization means different things to people. Here I'm plagiarizing a definition from a book that I found. I'll just read it out. The procedure of finding and comparing feasible solutions until no better can be found. Yep, that pretty much sums it up. That makes sense, right? Most real-world problems, you're going to deal with more than one objective, and they will be in conflict. Just imagine you want to optimize for the quality of a product. Well, you want to take into account how much that will cost, right? So they kind of contradict each other. Throughout the talk, I'll also talk about the knapsack problem, in which you have a bag and you're going to put in it packages of different weights and values, and your objectives are to maximize the value of the bag and minimize the weights. In order to have a fruitful discussion on this, you have to imagine two spaces of interest. We have the decision space. Those are the parameters that you have full control of. In the knapsack problem, those you make the decision of which packages to put in the bag. And then you're going to have a mapping to the objective space. That's the parameters that you're interested, the total weight and the total value of the bag itself. You know a topic is interesting if there's an illustration in XKCD. And so I like very much this subjective distribution of fruit. And you'll all agree with me that if we want to optimize for taste according to this, we're all going to choose peaches, right? And if we're going to optimize for how easy it is to eat the fruit, then we will give the author of XKCD, we'll offer him seedless grapes. But the point of this is, how are you going to do multi-objective optimization? You want to optimize for both parameters. So just a raise of hand, who thinks that you're going to opt for the peaches? Nobody. All right. Who's going to opt for the seedless grapes? All right. We have a few people raising hands. Any other ideas? What else are you going to suggest? What's that? I hear bananas. Okay, well that's kind of suboptimal. Well, you're from Colombia, so maybe that's why. Yeah. All right. So basically, another option is strawberries. If you go along a 45-degree line or something like that, take even weights. But actually, it was a trick question. According to multi-objective optimization, there is no one optimal solution, but rather a set of trade-off solutions that are equally optimal, and that's what we'll call the Pareto Front. I'll get back to this point later on. But let's kind of see what we were analyzing in our heads. And this is kind of what a lot of biology labs do around the world in other experimental environments. You have this subjective space. You want to optimize for 2 or more parameters. Throughout this talk, all the images will be in 2D, but take into account that you can extrapolate this to n dimensions. This is the space you're interested in, and you do your experiment. That's your flashlight in which you navigate in the space. The question is, what's the most efficient way to navigate? I'll present here 2 what I call 1D approaches. The first is called linear, in which you just maximize one direction, you maximize a second direction, and you say, okay, this is my optimal solution. You could alternatively go through this trajectory and choose this one. A second thing that you can do is what it's called single objective optimization, in which you come up with a metric that combines both objectives. In this case, I'm giving them equal weighting. We're going along this trajectory, and then we're selecting this as our optimal solution. But just looking at this, you can appreciate that this is really not satisfactory, right? There's a lot of space, a lot of unknowns over here because we designed our experiment in this fashion. What you would want to do is you want to turn on the light and look at the full objective space and create a full map such that when you're designing the next experiment, your domain expert, in my case a biologist, will point out, well, actually, I think our next experiment should focus on this area right over here. And that's the purpose of multi-objective optimization, in which you want to explore the full space, and that way you'll get close to the true optimal results. And again, what's the difference between the 2 approaches that I'm describing? In the 1D optimization, we set strong priors to the space that we're going to be searching. And in multi-objective optimization, what we're doing, we're making that subjective decision at the end. Or you can imagine, once you're familiar with the space, you can do it in a more interactive fashion, making decisions throughout. Again, back to the point of the fact that we're not looking, in multi-objective optimization, we're not defining one optimal point, but rather we're looking for a set of trade-off solutions. There's always going to be a trade-off. For example, over here, we have 2 objectives. We want to go over here, but what we'll have is we'll have a front of trade-off solutions that we call them equally optimal. Later, after we finish the search, that's when we'll decide which focus we're most interested in based on all the feasible solutions that are out there. So this front over here, it's also called the Pareto Front or optimal front, named after the economist from the 19th century. How do you mathematically define the difference between the Pareto front to the others? Well, the idea is that the optimal points, they're what's called non-dominated. That means there's no other point that dominates it in all the other dimensions. Let's take, for example, K over here. K is dominated by D both in objective 1 and in objective 2, as opposed to D, which is dominated by C in objective 2, but not in 1, and it's dominated by E in objective 1, but not 2. So there's no other point that dominates D in both objectives, and hence it's part of this optimal front. Another nice aspect of this is that we can be agnostic about the scalings of the two objectives. For example, if you imagine the knapsack case, you have weights and values. But what if you don't know how much the weight is worth in monetary terms? So you can be agnostic about that and make those decisions after you explore the full feasible solution space. I pretty much finished task number 1. Hopefully you have an appreciation for what multi-objective optimization is. What I mean by that is that you're optimizing not for one point, but for a set of trade-off solutions called the Pareto Front. And this will yield better results than 1D optimization because you're accessing a much larger search space. The reason you're doing that is because you're keeping the prior constraints, you're not setting prior constraints, but you're making your decisions after the search. So that's the first thing that I want to convey. The second thing is this applicable for your own projects that you're working on? I've been thinking about this, and so far I came up with this checklist of things that you'll be needing. The first is you'll need full control of your decision space. And what I mean by that is that, for example, I always imagine multi-objective optimization in terms of food delivery companies, right? They have 3 objectives. They want to satisfy the people at home. They have the restaurants, and they have the delivery personnel. And I was really excited when I first met for the data scientist from one of the big delivery companies here, and I wanted to know all the details about what they're doing. And then I realized in their particular department, they don't do multi-objective optimization for the reason that they have no control over the orders that the delivery personnel take. That's something that they don't have control over. So that's why I came up with this part of the list. The second thing is you want an inexpensive mapping between the decision space and in the objective space. For example, you'll want a model that you give it the parameters from the decision space, and it will immediately give you a result. If you have to go into the field and it takes time to get your results, then it might not work for that particular project. But basically, that's all you need. And once you have that, you create a map of the distribution of solutions, and then you go to your domain expert to design the next experiment. So it sounds very simple, but I purposely neglected one crucial fact, is what is the size of the search space that you're looking at. Most likely you have an intractable search space like the knapsack problem. The more packages you add, it increases factorially. And when we look at DNA sequence space, well, you can imagine, well, that's just something ridiculously large, and we need to use stochastic algorithms in order to address that. For that purpose, we find genetic algorithms are very useful. The reason is that it deals very nicely with combinatorial spaces, and it provides multiple solutions that are diverse. In the next few slides, I'll go over this diagram over here. This is one architecture of a genetic algorithm. In which the main operations, what you have is your solutions, you call them a population, and it's motivated by Darwin's theory of evolution, in which you have operations like selection, crossover, mating, which I'll get into. So first thing you have to do, you have to initialize your population. In the knapsack problem, you can imagine that you just have virtual knapsacks, and you just fill it with different packages. In our case, what we have is in this case, protein sequences. In red, I'm just highlighting differences from a protein that we know that works in the wild, something that we call the wild type. After you initialize the population, then you want to score for them. That's our objective. We want to know the fitness of each of them, and you start building these distributions. And then the next step is before your selection, you have to define a termination criterion. Can be like number of generations or reaching a certain region in your objective space. And then you want to select the population. For example, a naive approach would be, I'll just take my Pareto Front, right? My set of trade-off solutions. And yeah, that's actually a very naive idea because you might have only a handful of solutions over there that will minimize your diversity. And so to take that into account, there are various algorithms out there that will take all these things into account. For example, there's a thing called elitism that it takes into account, in which the selected group, they are the parents of the next generation. They will produce children via crossover and mutation. And just like if anybody's seen the movie Highlander, in which they can propagate to the next generation and they don't actually die. That's what elitism takes into account. And there's various interesting other aspects that are within these algorithms. It's beyond this talk. The next thing, the selected population, they're the parents of the next generation, and then you create children by crossover. In the knapsack problem, you can imagine just switching half of the packages. And in the case, and then, in sequences, what we'll do, we'll like chop sequences in half and just combine them. And in mutation, in knapsack, you just take out packages or put them in. And in the case of sequences, you'll just change one letter for another. In the next stage, so after you pass your turbination criterion, great. Now you have a list of a list of solutions that you didn't have before, and they're all scored for. And you have a mapping. You'll show it to your domain expert, and together you'll figure out, have you gotten the final solution that you need, or you want to go and do more experiments. And I'll get more into this point later. But for now, Zach, you kind of took the thunder away from me when it comes to animations, but I'll do my best over here to entertain you with a few. Here I'll just show an animation of doing this full loop, in which this is iteration number 1, and this is a toy model, and it's also a very non-realistic in the sense that I purposely made the objectives actually be aligned, in which there's not much conflict in it, just to show you what it looks like. You're maximizing objective 1, minimizing 2, X marks the spot, that's where you want to go. According to this, the Pareto Front is just this one solution, and just imagine this as a kid with a ball in the field and everybody's just trying to jump on it. So you can see this is really not real. This is what you'd hope for. This is the ideal. This is what you want. But in practice, what you'll get is something more like this. Maximizing both of them. You want to get to this point. Now you have like a much diverse Pareto Front here in blue, and all the rejects are the squares. And you'll get something like this, in which you see they're trying really hard to get here, but this whole region over here is probably not feasible according to this toy model. And what you'll end up is something like this, and then you'll sit with your domain expert, and you'll sit down and say, oh, maybe this is where subjectivity comes into, right? It's after the search, then you make the subjective decision. And you might select this one, for example. Great, so I just want to give a shout out to the developers of DEAP. It's a Python platform for prototyping evolutionary algorithms, hence it's very useful for multi-objective optimization, and they make life very simple. This is just a snippet of code that I wrote, in which I'm feeding their toolbox registry functions that I created for crossover, mutation, evaluation, and the most complicated one, which is the selection, I didn't even have to code myself. They have state-of-the-art selection functions that I briefly mentioned before. And I finished with task number 2, takeaway number 2. I gave a description of how to figure out if multi-objective optimization is relevant for your projects. You need control. You need full control over the decision space. You need an inexpensive mapping to the objective space, and if you have an intractable large space, you need a stochastic algorithm to search, and I talked about a genetic algorithm, which is useful to do so. Now I'll get on to what we actually do in LabGenius, in which we apply these ideas to protein design in order to make discovery of therapeutics much more efficient. We are a biotech company, London-based. We're in Bermondsey, which is south of the Thames. We've been growing really fast in the past year. I joined as employee number 12. Now we're over 30. here I'm showing our laboratory. We have, I think, over 15 biologists in which they are designing, building, testing for selection of various criteria of proteins. And in addition, we have automation engineers that are helping them having robots take over the repetitive jobs that they need to do. And they're also, the robots are taking over the night shifts. The biologists never had night shifts, but the robots are. And this is what our organization looks like from the point of view of the tech team. All the, here is where all the experimental design is, and they sequence the data, I mean they digitalize it, in which we clean it, we do machine learning, and the focus of this talk is library or protein design, and we do this in an iterative fashion in which once we're happy with the result, then we'll send it to our partner to develop a protein that we think will be successful to create a therapeutic. Proteins come in different shapes and sizes. Fortunately, they all have the common building blocks, 20 natural amino acids in which we can describe them in a sequence with just a character. This is a combinatorial challenge. If you take 2 amino acids, that's 400 combinations. Add a third one, that's already 8,000. It's not uncommon to get to 100, a sequence of 100. That's already more combinations than there are atoms in the universe. What does our data look like? I'll start with an apology because of confidential reasons. I'm not going to show labels on the data, and I'm actually not going to show actual data from our lab, but rather proof of concept that I did on public data. But everything that I'm describing is similar to what I'm applying on the actual data from our lab. So the data that we're looking at is, in this case here, I'm showing amino acid sequences, where again, the red is highlighting differences from what we call the wild type, something that we know works in nature, and it's scored for various protein properties. So this would be our decision space, and this is our objective space. The protein properties that we're interested in are things like stability for various screens, binding affinity, manufacturability. Then we apply machine learning, where what we're interested in is linking between DNA sequence to protein function. This is a whole talk within itself. I don't have scope for it right now. But the purpose of this slide is to show that if you have clean enough data and signal, you'll be able to create a reliable model. When we look at various data sets, we see accuracies of above 80 to like 95%, AUCs of above 0.9. And here I'm just showing binary classification in which we have out-of-box data looking at positives, clearly predicted to be as such. Okay, great. So the machine learn, the question is, how do we propagate these learnings to the biologist in the lab so they can design, build, and test for it? And for that, we build a sequence optimizer. And what it does, it takes the wild type, or it takes a protein sequence. That's our hypothesis of what we think will be useful. And then we'll ask the model for a prediction. But we don't only limit ourselves to that. We'll actually look at a whole large space of sequences and ask for their scores as well, and then we'll compare them, and then we'll suggest an improved candidate for either the next experiment or for the client. But of course, we're not going to look at one function at a time. We're talking here about multi-objective optimization. There's a whole list of things that we'll be interested in, and so here I'm just showing 3, but you can imagine more functions that we're asking for the scores, and hence we're applying the multi-objective optimization in order to get an improved candidates and make sure that we go over a full list of functions. Now I'll show a few slides in regard to a case study that I've been working on. The data here is binding of antibodies. They are 200 amino acids in length, but in the data there's actually only 20 locations in which are mutated, but that's enough to get a search space of 10 to the power 26. So clearly something that you want to use stochastic methods with. And just to describe this visual over here, this is what I call the frequency matrix. This is what the data actually this is one way of representing the data, in which here's the 20 positions. And in position number 1, for example, 90% of the sequences are T. And that's kind of the wild type. That's what we know actually works in nature. This sequence here is what works in nature. And then you can see the rest of the 10% are distributed amongst the other 19 amino acids. So that's the data that we have. We built the model based on that. And then we want to tweak these a bit and suggest different sequences than this wild type and see if we can find one that performs better. And this is an animation that we see, this animation is from the genetic algorithm. It's going through the decision space. And the way to read this is before I showed here the frequency, and here I'm showing the change in frequency. And the way that I read this is that over here, for example, where it's 0, it agrees with the previous frequency, so it prefers here the wild type. And all these just show that the wild type is being rejected. So over here, Y is being rejected, and it prefers S, sometimes K, and so on. Just to show you the initial distribution, here you can see half of the time I'm using the wild type, and half of the time I'm using at random all the rest. So I select this very randomly, and then that's when the machine learning kicks in. Machine learning is already set, and I'm asking for scores from it, and then it's pulling in different directions. That's what the decision space looks like. What does the objective space look like? I'll just rewind this. Sorry. All right, there you go. So you can see that it quickly started over here. The objective is to get as close as possible to this point, like over here. I should have marked it with an X. And you can see the Pareto Front pushing its way over here. The red squares are the rejects. The selected are in red. And when the selected becomes the final Pareto Front of the 160 iterations, that's when it turns green. That means it's not learning anything in that region anymore. And another thing that I added, which I like, is what I call the graveyard. Those are the ancestors of all the current population in each iteration. And that's useful to preserve so you can do analytics later on. Great, so you ran multi-objective optimization and you create this map. What do you do with it? You go to your domain expert, in my case it's the biologist, and well, they have to decide what they're going to select for the next experiment. If it's something that we're going to give to our partner, or it's something that we have to go back to the lab and find better solutions. And in this case, we just highlighted, well, this is where we want to go, but there's no feasible solutions over here, and so we have to do the next best thing. We just carved out solutions over here, and that's why you see these streaks. Actually, I wanted to go back and figure out more sequences over here so we can go back and test for these. Another nice aspect of this is the fact that we go further away from the wild type, which is done in regular, in standard biology labs. Just to explain what that means, what we measure is the Hamming distance from the wild type. And the training data is here in gray. So you can see the training data goes until a Hamming distance of 3. Normal laboratories, what they do is they don't go too far away, maybe 4 or 5 amino acids away from the wild type. And here what we show that according to our machine learning model, we can easily go to about, out of 20, we can go to a distance of about 14. And that's what you saw in the decision space. You could see it went very far out. And this subset selection is here in pink. All right, just checking on time. So I have a few more slides, which I'll breeze through, but that's pretty much what I want to describe. There are various other aspects of more details in multi-objective optimization. It's a really fun topic. And things you have to worry about is, for example, it's the diversity of the population. You have to worry about local minima. You're dealing with a stochastic algorithm, so how do you deal with that? And it also has its own hyperparameters. How do you even do tuning for that? So the answer to all of this is, first of all, you want to visualize as much as possible. In order to do that, you want to log everything so that you can do your analytics. These are just a few visuals that I find useful. For example, per iteration, I'm looking at the objectives of the selected in green and the rejects in red. Here I'm looking at the diversity. My population here I chose is 20 individuals, and I make sure that's a straight line. If I have duplicates, then it will quickly drop, and that's horrible because that means that all the children are based on the same parents, and you know that's never a good idea. This is an analogy, this sin x squared of the problem of local minima. So imagine that you start around here, then you'll end up over here. What you want to do is you want to do niching. That means you want to start in various locations. That way that you can look at the final results and get all the minima. Another thing that you want to do when it comes to the fact that you're dealing with stochastic results, well, you just want to, the simplest thing is just do many seed runs because you'll always get a Pareto Front when you combine the results of different runs. For example, over here, here's 3 repeats, different color codings, where in the triangles are the Pareto Front when I aggregate everything together. So in repeat number 1, it actually has 38 here marked in blue, but most of them are X's, meaning they're being rejected by the final Pareto Front, and only 5 of them contribute to the final Pareto Front of 30 individuals. Just because it's fun to watch these animations, this is what a similar result looks like when I stack together 10 seed runs of 2 different configurations, 5 repeats each. So it's always cool to find out and learn like how the evolution took place. That's all I had to say about multi-objective optimization. Just one short community announcement. We are looking for a bioinformatician to help us analyze our new generation sequencing data. If there's anybody in the audience who actually knows what I just said, great, talk to me. We're hiring. If you don't, that's fine. In a few months' time, we'll probably be in the lookout for data scientists as well. Just to summarize this talk, hopefully you all understand what protein design by multi-objective optimization actually means by now. I talked about multi-objective optimization in the sense that you're optimizing not for one solution, but for a whole set of trade-off solutions that are equally optimal until you apply your weighting after the search. And the reason that yields better results than the standard 1D optimization is because you're exploring a much larger space. I also give highlights of indications of why this might be useful in projects that you're working on. If you're dealing with conflicting objectives, which is most likely the case, if you have full control over your decision space, and if you have an inexpensive mapping to the objective space. And I also talked about how in LabGenius we're applying the concept of multi-objective optimization to make discovery of therapeutics much more efficient. I will just finish with a few resources which I found very useful in order to learn this topic. Basically anything that Eckhard Zissler wrote since his PhD, I highly recommend. For prototyping, I recommend DEAP, and yeah, all the animations, just matplotlib, and I do appreciate how frustrating it can be, and I did come up with my own mechanism of doing it fairly quickly now. Yeah, but thank you very much.
Thank you, Eyal. Yeah, we have time for a couple of questions.
Yes.
Thank you for a very good talk. So a few questions for me really is, the first one is, because I think a lot of people here are more familiar with derivative-based optimization techniques, right? Using, so how is this, if you have 2 objectives, let's just stick to 2D, you have 2 objective functions, then each of them are functions in their own right, right? So it means this decision space is like a function composition, if that's...
The decision space is what?
It's a function composition, right? You have 2 functions together going into another function, right? Is my reasoning correct? So I'm trying to see if I actually understand what's going on here. So you have, what does the surface actually look like, the decision space, if you plot it? Does it look convex? Is it non-convex?
I see. Okay.
Right. So that's the first question. And are there people that use, derivative-based techniques for this? Like
do they use what?
Derivative-based techniques, like maybe gradient descent or any of those other more familiar, techniques people are more familiar with. So
yeah. Okay. Excellent questions. So hopefully I understood both of them. So the first one has to do with what does our decision space look like, and the second has to do with should we just be applying gradient descent.
yeah, could one use that?
Yeah, so in our case, and imagine also in the case of like the knapsack, it's combinatorial. And so that means that any change, like if you take one package out or in, that you have like a very choppy landscape. And that's the reason why gradient descent is the best choice for that. Yeah, I think that kind of summarizes both of your questions. So you can use gradient descent if you know you have like smooth decision spaces. But once again, combinatorial, yeah, you kind of want to go with multi-objective and if it's very large, like genetic algorithms. Yeah, and back.
Sorry.
Yeah.
Maybe now. Okay. So I just had a question about how you do the multi-objective optimization. I was just curious, I don't know if this is a stupid question, but after doing this iteration, on top of understanding things about the protein sequences and so on, can you also understand, for example, like that maybe some of the parameters are not that important, or you can limit the parameter space of one of these parameters so that, you know, maybe in the next iteration it can be done more quickly, or you understand something more about those parameters?
Yeah, so your question is about the parameters in the decision space deciding if they're important or not. Yeah, so that's a very good question. That's something that I haven't got into. It has to do with optimizing your optimizer. How do you know if by tweaking a few parameters or excluding part of the decision set if you're doing better or not? Yeah, that's a whole subject within its own. The short answer is that there are metrics to compare Pareto fronts in order to figure out contributions of different hyperparameters or different. Actually, that's a good question. How do you compare? Yeah, I'll really have to think about that. Maybe catch me in the pub later. We can brainstorm that out, because I thought I had the answer and I started saying something, but I see that I got to a roadblock here. Yeah, but catch me later. Yeah. Yes.
Hi, is it working? No?
No, but I
No.
hear you. I'll repeat your question.
For the rest,
Yeah.
I was thinking about, are the different parameters independent of each other, or, for example, one parameter actually influences another one?
Mm-hmm.
And I was thinking that maybe you would have a protein that is not on your front, but it's a little bit inside, like in the graveyard, but it has some synergistic properties that the combinatorial of different values on your parameters will make that the final effect when you're testing on the lab will be much greater than what you are expecting from looking at just the front.
So the question is, sorry, could you repeat that?
Are you trying to are you only looking at the front, or are you considering some proteins that are inside that area that will have some synergistic effects because of the different combinations?
I see. So the idea is, so your question is, when I do the optimization, do I consider only the Pareto front for the next iteration, or do I go back and look at
Yeah, something like that. Because, for example, if your final objective is to, let's say, have an enzyme that has certain, well, enzymes are very unstable.
Mhm.
So, for example, if you will change a couple of states in the sequence, that will make it much more stable. But maybe that protein is not on the front; it's much more inside of you.
Yeah. Okay. So I'll try to answer it in the sense that you want, in the case of proteins or the knapsack problem, you always want diversity. The fact that you rejected something in the past, like you might want to take aspects of that later, and that's why you want a diverse, in the terms of the decision space, you want a large diverse population, so that if part of it is rejected, in the sequence space, maybe that part might be picked up later on. I don't know if that answers your question. Yeah, but I think the key is diversity. Is even if something has been rejected, parts of it might be useful later on. And again, like in the example I showed, it's a factor of 10 to the 26. One aspect that I didn't mention, as the optimizer, it currently like treats machine learning as God. So like even if you have a crappy model, you have a crappy optimization. That goes without saying, but one has to always have that. Take that into account. That the quality of the search depends on the quality of the machine learning. Yes.
One last question.
Yeah, so just wondering, you were saying initialization is very important. For your toy problem, I don't know if I missed it. How did you initialize your protein sequences in that toy problem?
In this case study, I did the naive thing of just taking the wild type. The reason being, the wild type is something that we know works in nature. And if you take something like, imagine this is a combinatorial space of 10 to the 26. If I would come up with a random sequence, then I would really rely heavily on my machine learning to have realistic biological predictions, as opposed to keeping close to your training data. So I showed that I managed to go like 15 amino acids away from the training data, but if I had like training data with variances of like 100 in length, I seriously doubt that that I get reliable results if you go like too far away. Like you do want to stay close to the wild type, but that's something that we're still exploring. Yeah.
Okay, let's wrap it up. Thanks, Eyal.
Okay, thank you very much. I'll be in the pub for more questions.